It's a real pleasure to welcome Galit Lahav, who's coming to us from Harvard Medical School, and she's been there for 22 years and has been department chair since 2018. And obviously, systems biology and those approaches have a lot of overlap with mathematical oncology. Obviously, our focus is purely oncology, but the idea of using sort of model-driven approaches to better understand those complex systems is really at the heart of what we do. So it's a really great pleasure to have Galit here and for her to give us our opening plenary. So please.
Thank you, Sandy, and good morning. Pleasure to be here. First of all, it's just beautiful. It's already becoming really, really cold in Boston, so it's nice to escape to this warm weather. I'm excited to share with you some highlights from the last 20 years, and I would start by saying that we are a lab that doing mostly basic research, fundamental research, and to me to come to a conference like this is a great opportunity to think how some of the insights we develop, mostly in tissue culture cells, a little bit in vivo, I'll show you at the end, can be utilized to the things that you guys are thinking in terms of evolution of cancer and treatments and resistance. And so I'm going to talk with you today about information transfer through protein dynamics. And basically, I'll start with a super basic question that we have in the lab, which is how cells read instructions, right? How cells know that in response to a specific stimulus or input, it's time to divide or to arrest or to move or to activate cell death. And the simple answer to this, and as we know from many years of molecular biology, is that information is transferred through what we call signaling molecules. And it could be as simple as the amount changes of signaling molecules. Then you have a signal that is increasing in amount. It can be the identity, meaning that a molecule is being modified, phosphorylated, methylated, or it can be localization, that a molecule moved from the nucleus to the cytoplasm, right? Or more complicated than that. And in our lab, we are thinking about another layer that talks about information transfer. I'm not sure many of you are thinking about that as well, which is the dynamics of signaling.
Stand the mic, or you can take this if you like to walk out.
No worries. I'll try not to move. It's hard, but I'll try. The reason is that when you look at things at a snapshot, we just look at a snapshot, it can mask different dynamical responses. And what do I mean by that? Imagine that you want to study the mechanism of action of 3 drugs, and you measure the level of a molecule, whatever the concentration, modification, localization, and you see that before you added the drug, the level was low, and after you added the drug, the level was high, okay? And this is how it responds to drug A, to drug 1. And now you're looking at drug 2 and drug 3, and you see exactly the same thing. Before the drug, it was low, after the drug, it was high. You may conclude that the mechanism of action is similar between these 3 drugs. But if you have tools to follow the behavior of this molecule over time, to follow the temporal dynamics, then you may discover that the behavior is very different. In response to drug A, maybe it went up very quickly and then plateau. Drug B, it stayed low and then maybe switched, and drug 3, it oscillated. Okay, so very different dynamical behavior, telling us mechanism of action of these drugs are different. And so by looking at protein dynamics, you can actually understand how different inputs are encoded in the dynamics of signaling molecules. And by I mean encoded, it can teach you about the network structure that control these signaling molecules. And you can also ask questions about how these are decoded. How do you get specific cellular outputs in response to these dynamics? And I want to stop and say that the way we think about signaling dynamics is like a target, like a drug target. We usually think about a drug target, a small molecule that inhibits the function of a protein. We think of the dynamics as a target. If different dynamical behaviors mean different things to cell or give different information, then you can target the dynamics, you can change these dynamics and control the decisions that cells are doing. And once you start to think about dynamics, it is way more rich and way more complex than just level, localization, modification, right? You start to look at all these parameters related if something oscillates to a frequency or amplitude, integrated level, delay, and so on. And we are really obsessed with understanding how input and code in these different parameters and how, in what ways cells have to read these parameters in order to execute the right cellular outcome. And because many times individual cells show different behaviors, we perform our studies as much as possible at the single cell level, right? And I think especially also when thinking about resistance to treatment, there are many situations that we know that if we take even genetically identical cells, we treat them with radiation or with drug, we see that some survive and some die and some escape. And these that survive are the ones that can later become resistant. And this is even without any genetic differences, just different behaviors in response to the same treatment. And so as much as possible, we say to understand fundamental processes in biology, as well as why a subpopulation of cells escape drug actions, we investigate the dynamics of cellular responses at the single cell level. Okay, these are the two things you need to remember: dynamics and single cell. And with this big umbrella, we in the last 20 or so years have looked at multiple different pathways in human cells: regulation of cell cycle, cell growth, apoptosis, differentiation, DNA damage and repair, also recently oncogenic activation. But the pathway that has been the focus of the lab and kind of like the thread of the lab all these years is the p53 signaling pathway, which I'm going to talk with you about today. So I'm sure we all know that p53 is a tumor suppressor, that about 50% of human cancer have mutation in p53. Of course, depending on the different cancer, this number changes quite a lot. And it's what we call the hub. Okay? It's a protein that responds to many different inputs in the form of some kind of stress. We focus on a stress in form of DNA damage, mainly response to radiation or chemotherapeutic drugs. And then p53, a transcription factor, assess the damage, assess the stress, and activate different targets, the transcription of different targets that are in different transcriptional programs. And some of these programs mean repairing the damage to transiently arrest and then recover. And some of them are what we call terminal fates, like a permanent cell cycle arrest, senescence, or cell death, apoptosis. And a big question in the p53 field has been how this single molecule, the single protein of p53, make the choice between these different outcomes that, if you think about them, they're a little bit conflicting. Some of them mean recovery and survival, and some of them are terminal, and there's no way to recover. And this question is really important, not just to fundamentally understand how p53 acts as a tumor suppressor, but also there's a big effort now in the cancers. Remember I told you 50% have mutations in p53, then 50% of cancers don't have mutations in p53. They have wild type p53. And there's big effort both in academia and in pharma to find ways to activate, wake up this p53 and control its function in order to push those cancer cells toward terminal fates, such as apoptosis and senescence. Also, in cancers with non-functional p53, there is effort in putting wild-type p53, restoring p53. And again, we want to better understand how can we just not like throw a ton of p53 on cells, but really carefully change and control its dynamics so that we can push cells into the correct outcome. And so many years ago, my lab has developed tools to follow the dynamics of in individual cells. And before showing you the results, what you need to know that is critical about p53. Oh, first of all, I'll tell you what I'll show you in today's talk. I'll show you that we found about p53 in response to DNA damage. For this conference, I decided to focus on a few examples from the last few years in which p53 dynamics help us guide therapy, again, mostly in cell culture, a little bit in vivo. And then I'll show with you a more recent study from this year, the effect of cell confluency on p53 dynamics, and a little bit of studies we did in vivo. Okay. So what do you need to know about p53 regulation? P53 expression, the mRNA of p53 is constant, and it does not depend on if a cell has damage, if a cell is stressed, or if a cell is happy. It is constantly being transcribed. And then p53 activate the transcription of a protein called MDM2, which is an E3 ubiquitin ligase. And MDM2 bind p53, ubiquitinate it, and send it to degradation. Okay, so this creates this very classical, what we call negative feedback loop, where you have one transcription arm, which is a slow rate response, and one degradation arm, which is a fast rate response. And you can play with these two parameters, and you can get a bunch of different behaviors for p53 and in response to DNA damage. And what we found several years ago using live cell imaging and fluorescent reporters with p53 is that if you take human cells, and it can be cancer cells or healthy cells with wild type p53, and you irradiate them with gamma radiation that creates double-strand breaks in the DNA, then cells go through these series of really beautiful oscillations that can persist for many hours and even days. If you look at the individual cell level, these oscillations are undamped. Okay? They don't go and become smaller. They have the same amplitude. The amplitude is a little noisy, but on average, it is the same. And the period is extremely robust. The period doesn't change. It's a period about 5.5 hours. It is extremely robust between cell line, between cells in one cell line, between different doses of radiation. Very difficult to change the period. And when you look at the individual cells and average them together, it looks like that oscillations are damped, but they're undamped. We also can look at p53 together with DNA breaks and can see, it's a little hard to see, I think, the colors here, but the green here is p53. The red is a marker that binds to DNA breaks. And you can see p53 pulsing, and then the number of double-strand breaks go down with time. It's a little hard to see the green, but you can see that the red is going down. You get less and less and less DNA breaks. And so we realize that this system behave like a while loop. They are pulse of p53. Then it says, while there is still DNA damage, do another pulse, right? Then pause, check, is there still DNA damage? And if the answer is yes, give another pulse. If not, stop pulsing. There's, again, no damping. It just pulse and then stop pulsing. And one of the things that we were very interested for a long time is to understand what these pulses mean, why p53 oscillate. And we had a great tool to manipulate these dynamics using a small molecule called Nutlink3, which is an inhibitor of MDM2. And by adding Nutlink3, we can perturb the interaction between p53 and MDM2 and then eliminate these oscillations. Now, that turned to be not a very simple task because this is a dynamical system, and you add more Nutlink3, then you get more p53 that gets more MDM2 that compete with Nutlink3. And this is where we went back into a model, a kinetic model, that it will allowed us to develop a protocol of how much Nutlink3 we need to add at which time point in order to eliminate p53 oscillations and lock them into what we call a sustained level. And then when we compare p53 oscillations with sustained p53, we found through a series of studies that just by changing p53 dynamics, we can change the kind of genes that p53 activate, and we can change the final cellular outcome. So in response to p53 oscillation or to pulse p53, we mainly get DNA repair, recovery, and growth. But if we switch p53 oscillations into sustain, we get senescence and apoptosis. Okay? And we looked at both at the transcriptomics and proteomics and binding to DNA in order to also understand the mechanism mechanisms that connect p53 dynamics with cell fate. Now, in this study that was done already many years ago, we did a very big change, right? Oscillatory and non-oscillatory. And since then, we've been working really hard to see if there are ways that we can do a bit more smaller perturbation. For example, remember I told you the period is so robust, this 5.5 hours. Can we change the period and see what happened? And only recently we found a way to do that. We have a recent story in which we showed that p53 oscillations in response to radiation can be reset and can be entrained to an external stimulus. If we provide periodic addition of Nutlink-3 in a microfluidic chamber, we can irradiate cells and they oscillate, and then we add Nutlink and we can have p53 entrained to this new period and then remove Nutlink and go back to the other period. Okay, so now we have a system that we can look downstream of how frequency modulation of p53 impact gene expression and cell fate, and this is in progress. In a few years, I might be able to share that. But I want to share one more thing related to that because I think it's really cool, and I don't fully understand that. We also saw the p53 oscillations, they show resonance at the natural frequency, meaning that if we remember I told you we radiate cells, the frequency is 5.5 hours. Now we can use external stimulus, okay, to change the frequency. And when we do that at lower frequency, 5.5 hours, and higher frequency, we see that at the natural period of 5.5 hours, the amplitude is the highest. Okay, we get lower amplitude at lower frequency, lower amplitude at higher frequency, but at the natural frequency, we get resonance. We basically get enhancement of p53 amplitude. We will feel like this is pretty cool. Again, we're trying to understand what is it about these 5.5 hours that cells evolve to oscillate with this frequency, which is not anything related to cell cycle, circadian clock, and so on. It's very unique frequencies we try to understand. Now, this is all focused on oscillations, but we know that if we use other kinds of DNA damage, we can get different dynamical behavior for p53. So if gamma radiation gives oscillation, if we use UV radiation, we basically get just one big pulse of p53. If we use cisplatin or other carboplatin drugs, we get an increase in p53 in apoptosis, and I'll show you some data for that. And if we do a knockdown or inhibition of MDMX, which is another oncogene that suppresses p53, we get these biphasic response, one big pulse, and then oscillations. Even when cells proliferate in a happy, healthy conditions, we found what we call spontaneous pulses of p53. We see that every once in a while a cell show one pulse of p53, and then that's it. It's almost like the guardian of the genome is kind of coming to check everything is okay, and then becomes quiet again. So I want to show you some examples related to cisplatin and MDMX, in which, as I said, looking at p53 dynamics can guide the design of combinatorial therapy, at least for right now, at least in a dish. This is a movie of cells expressing a p53 reporter in response to cisplatin for what we call the IC50. And what you see is p53 levels. You see this greenish yellowish color. This is cells starting to activate p53. And in IC50, as you know, 50% of the cells die, 50% of the cells survive. So we said, okay, let's look at what p53 is doing, right? I find it really fascinating to even IC50 understand, like, what is it? Why 50% die, 50% survive, right? And we know that p53 plays a major role in this decision. And if you look at the final picture from this movie, it's messy, right? The biology is messy. You see a lot of dead cells here. You see cells that survive with relatively low levels of p53, but then you see quite a lot of cells that survive with very high levels of p53. And so how is this possible, right? The canonical model was that if you have a little bit of p, you'll survive, but if you have a lot of p53, you will die. So when we extract p53 levels from hundreds and hundreds of cells, and showing just a few examples here, we see that single cells show different rates of p53 accumulation. Some cells accumulate p53 very quickly, and some accumulate it slow. And if we color them according to whether a cell died or a cell survived, we see that on average, dying cells accumulate p53 much faster than surviving cells. Okay? And now we want to find a threshold, to say a threshold which above that level a cell will die. And we try to put a threshold and it just doesn't work. There's no threshold that we can put that will fit whether a cell die or survive, right? If we put it too low, then we have a lot of surviving cells that actually cross that threshold and they still survive. And if we try to move it, again, we have a lot of dying cells that are below that threshold and they died. And through a lot of live cell imaging and statistics, we found that we have what we call a dynamics threshold. There is a threshold for p53 levels to activate apoptosis and die, but the threshold moves with time. And with time, you need more and more and more p53 in order to execute apoptosis. And again, through a lot of literature reading and checking and screening, we found what is the mechanism leading to this time-dependent threshold. And the reason is that in response to cisplatin, we're not just activating p53, we're activating a second pathway, the pathway of IAP, which is inhibitor of apoptosis. Okay? And these two pathways basically compete with each other. So if p53 goes up very, very fast, right, then it wins and it kills the cell. But if it goes very slow, in the background, you have this inhibitor of apoptosis that is induced. It competes with p53, and now you need more and more p53 to execute apoptosis, which can explain why we have this time-dependent threshold. And so how can we change that? There are two combinatorial therapies that we can do to change that. First of all, we can add NUTLINK3, right? This is the MDM2 inhibitor in order to accelerate p53. So we lead p53 goes up faster in all the cells. And now in the concentration of IC50 plus NUTLINK3, we get 84% cell death. Another way to do that is basically block the IAP pathway. We, at that time, we were working with Novartis, and they just developed this small molecule that inhibit IAP, it's called LCL161. And when we block this competing pathway, the threshold now become a static threshold. Now, all cells care about is p53 because there's no more competition. Once p53 crosses the threshold and the threshold stay flat with time, then the cell will die. And now with the same IC50 and this small molecule, we can get to over 90% cell death. Okay. And I would add also in this study, we played with the time between cisplatin and the small molecule that inhibit the IAPs. And we realized that in order for this to work, we need to add them at a certain time interval. If we add the inhibitor much later, like 24 hours later, it doesn't matter, okay? You can add it and you don't change the concentration or the percentage of cells that die because the IAP protein already activated and the pathway already compete with p53, okay? I want to show you another example in which we show that the timing matter in terms of combining two treatment. And this is where we focus on the role of MDMX, I mentioned earlier. MDMX is an oncogene that suppresses p53, and a relatively high percent of cancer, there is overexpression, amplification of MDMX. So there is quite a lot of effort in developing small molecules that will inhibit MDMX, again, for those cancers that have wild-type p53 in order to allow it to use the wild-type p53 to kill those cancer cells. And using reporters for p53, we found several years ago that if we inhibit MDMX, we create these biphasic dynamics. Cells show one big pulse of p53, and then a series of oscillation. And I have to say about this assay, many times the heterogeneity between cells is very big, like different cells show different things. In this particular assay, it was amazingly robust. We added the MDMX inhibitor, and all cells show this one big pulse of p53 and then a series of oscillations. Okay? In this study, we looked at what each of these p53 in different phases is doing, but I'm just going to show you the final conclusion that we found from that. We were wondering if we take this two phases of p53 and combine it with DNA damage. We combine MDMX inhibition with DNA damage in phase 1 and in phase 2, right? Phase 1 is this one big pulse. Phase 2 is this series of oscillations. How this is going to impact, how this is going to synergize with DNA damage, okay? And what we found is that if we add DNA damage at phase 1, then we get a synergistic effect. MDMX suppression synergizes with DNA damage at phase 1. We get way more cell death than without MDMX inhibition. But if we do it at phase 2, when p53 oscillates, we don't just make it less synergy, the synergy less, but we flip the interaction. Now the interaction become antagonist. And now when we do MDMX inhibition, it antagonizes DNA damage at phase 2. This is happening because at phase 1, most of the apoptotic genes are induced, but in phase 2, cells activate cell cycle arrest, which we know protect them from apoptosis. So basically, you have this situation in which you added one molecule that inhibits MDMX, it creates this very complex dynamical behavior, and now the interaction with the second drug, a DNA damaging drug, flip depending on the time interval between the two of them, right? Again, this is a study developed now almost 10 years ago. We were hoping at that particular time that this will inspire us to some trials or work to really look in vivo in patient about this combination and other combination. I feel like this hasn't moved into this direction, and happy to use this conference to talk with anyone here to think, how can we actually make this happen for this study or some of the other studies that we have. But somebody in related to that, one of the things that we've been asking ourselves in the lab quite a lot more recently is how well do tissue culture reflect real physiology? Right? Everything I've shown you so far is we take cells in a dish and we treat them, and cells in a dish are very, very different than cells in our body. And so in the lab, we're trying to develop some new experimental systems to study the response to DNA damage and study p53. We compare 2D versus 3D. We're looking at tumor spheroids. We're looking at organoids. Really beautiful studies, really complicated, but it's rich and in some ways behaves similar to cells in a dish, in some ways a little different. Interactions between cells, we're really interested in interaction between cancer cells, immune cells. I'm excited to see there's some talks here about these topics. I'm looking forward to that. Cell confluency, again, we're growing cells in a dish. They're all happy. They have a lot of space to grow. Not exactly what is happening in our body. And in vivo, look both at what's happening in normal tissue and in xenografts. And so I want to start by talking about cell confluency, and this is a recent project done by two postdocs in the lab, Alina Simerzin and Emily Ackerman, in which we just ask a very simple question. Does the level of confluency affect the response to DNA damage? And we took cells at low or normal confluency. This is what we usually do our experiment. And then we grew cells at very high confluency. They're dense. They have no way to move. We irradiate them and then compare the behaviors. So for low confluency, we got the same oscillatory behavior that I described to you throughout the talk. This is example of 3 single cells. This is the population. Seem like it's damp because they lose synchrony. But for high confluency condition, we got a completely different behavior. We got, instead of these series of oscillation, we get one big pulse of p53 and then some small fluctuations. Okay. And so if we look at the different parameters between these two behaviors, and here the low will be gray and the high confluency will be red. We see that the duration of the first peak is much longer in the high confluent condition. The timing of the first peak is delayed. It happens later, it's longer, and the amplitude is a bit higher. Okay, so totally different shift in dynamical behavior. And so we wanted to figure out, okay, what can explain this different dynamical behavior in high confluency versus low confluency? We have a model that we developed for P53 many years ago. We said, okay, let's go back to our kinetic model and try to use the model to tell us which parameters are the most sensitive ones that can switch oscillatory behavior into this signal pulse that we see. So we perform a global sensitivity analysis to tell us which parameters the model is mostly sensitive for, and two parameters came up from this analysis. The MDM2 mediated P53 degradation, quite intuitive, not surprising. And the second is WIP1 degradation. Let me just tell you what is WIP1. WIP1 is the second feedback that responds to P53. It's a transcription target of P53. It's a phosphatase that dephosphorylate P53. It also dephosphorylate the upstream kinase that activate P53. So it creates a second feedback on P53, okay? And so MDM2 mediated P53 degradation, we've studied this quite a lot. This is if we do in the model, we see that lower degradation of P53 by MDM2 eliminates the oscillation, again, intuitive and kind of nice to see in the model. And as I showed you earlier, that's something that many years ago we already explored and checked how different concentration of Nuttling-3 at different time can eliminate P53 oscillation. So we were kind of, okay, this one we know, let's put it aside and let's explore the other parameters that came from our sensitivity analysis. And this is the higher degradation rate of WIP1, which again, just by simulation of the model, is also predicted to eliminate P53 oscillations. And so let's go to the back to cells and see whether indeed in high confluent conditions, they have less WIP1. And just through a Western, we see that this is really the case. This is low confluency. This is WIP1 levels pre-DNA damage, and the levels increase after DNA damage. And in high confluent conditions, we have much low levels of WIP1, both before we irradiate the cells and also after irradiating the cells. So this is just correlation, right? That shows us that indeed high confluent conditions have low WIP1, and in high confluent conditions, P53 doesn't oscillate. The model says these two things are connected, but we also need to prove it experimentally. And again, we use a small molecule that inhibit WIP1 and look at P53 behavior in low confluent conditions where it would usually oscillate. And what we found is just inhibiting WIP1 had a very strong effect on P53 dynamics. This is regular low confluency. The black is regular low confluency. The red is high confluency. And this is if we do low confluency plus WIP1 inhibitor, we eliminate these oscillation and get this one big pulse that resemble the behavior in high confluent conditions. If we look at the different parameters, we see that WIP1 inhibition is doing a pretty good job in terms of switching it to behave like high confluency. It makes the first pulse wider, longer. It makes it delay, but it doesn't have much of effect of the amplitude. So it can explain most changes of dynamics, but not all of them. And this is, again, where modeling can help us, because now in the model, we can perturb both of these parameters. We can change the MDM2 dependent P53 degradation, and we can change WIP1 degradation rate, okay? Our model that leads to P53 oscillation uses the parameter in this little square here, right? So in 24 hours, we get about 4 pulses. This is the timing of the first peak, and this is the first peak amplitude. And then we see if we just move on this axis of WIP1, we can change the number of pulses, the timing, but we don't really change the amplitude. If we just move in this one, again, we can change some of the parameters, but not all of them. But then there is a region here in which we change both WIP1 degradation and MDM2 dependent degradation, and we can get to the behavior that we see in high confluent conditions. Now, why does this matter? It matters, first of all, because a question is, what is the real confluence see in a mouse, so in a human, tumors are often considered to be more dense than normal tissue. Does this mean that we will get different dynamical behavior in the tumor versus the rest of the body? This could be a good thing if we want to protect normal cells and destroy cancer cells. So this is all something that we're looking further into, both in vitro and in vivo. And I want to show by just showing, finish by just showing you a few studies in vivo. We're going to leave a few time for questions. So this is a study that we conducted in collaboration with the MGH Center for Systems Biology, the Galit Lab. So first of all, what you need to know is that p53 oscillates faster in a mouse. Remember, I told you that the frequency of p53 in human cells is 5.5 hours, very robust, can't break it. Now we have new tools to do it, so we can see what it means. But if you just take mouse cells, mouse and rat, it oscillates faster. And not only it oscillates faster, it oscillated like half the frequency. If human is 5.5 hours, in mouse, it's about 2.7 hours, okay? Again, why? I don't know. Like, we understand mechanistically why this is happening. Like, we can take a p53 from a mouse and put it in human cells. How do you think it will oscillate like a human or a mouse p53? Like a mouse? Well, the thing that matters is the background. So if you take a p53 from a mouse and put it in human, it will oscillate like human p53. If you take a human p53 and put it in a mouse cell, it will oscillate in the frequency of the mouse. So it's not the sequence of p53 that matters. What matters is the system it's at, and we were shown that it's actually an area in the promoter of MDM2 that is different between mouse and human that control that frequency, okay? But what it means is that, I think about if you have a xenograph in a mouse that is taken from human cells and you irradiate the mouse, the p53 in the xenograph will oscillate in p53 in the rest of the mouse will oscillate in different frequencies. So just something to keep in mind, I think, when we do this mix and match experiment. But anyway, in vivo, we have not been able yet to create a live cell reporter for p53. So all the data that we collected in vivo so far is using fixed tissues. So we basically took a mouth, irradiated the whole mouth, the whole body, and then extracted tissues from the mouth and use immunofluorescence to measure p53 levels in different tissues. Okay? This is how the data looks. I love showing this picture just because it's very, very pretty. Small intestine. This is 240 images stitched together. You can see red is DAPI and the green here is p53. You can also see different that the localization, that cell type affect p53 induction. We see it way more in what's called the crypt of stem cells, which is just something that we are following up now using intestinal organoids. But if we look at a bunch of different tissues in the mouse, we see that in response to radiation, p53 is induced across tissues. Again, this is using immunofluorescence. We look at thymus, spleen, large intestine, small intestine. We get induction of p53 levels if we compare with no radiation versus radiation. Okay, green is p53. But now if we look at the dynamics of p53, and again, remember, we can't really do it now at the single cell level over time, and we can't just look at a high temporal resolution. We can take every hour and look at the entire population. But what we see is that overall, radiosensitive tissues, which are tissues like thymus, spleen, bone marrow, they showed a sustained level of p53. They induced p53 and then p53 stayed flat. Radiosensitive are the ones that show mostly apoptosis in response to radiation, while the radioresistant tissues, small intestine, large intestine, and skin, show a more transient induction of p53 or something that may even resemble oscillations. Now, this fits really beautifully to what we see in cell culture. Remember that the oscillations were correlated with recovery. Sustained p53 was more leading to apoptosis and senescence. So now we see these natural variations between tissues and the dynamics of p53, which also correlates with their known sensitivity to DNA damage. Again, we don't think that everything is P53 and all radiosensitivity comes from P53 dynamics, but there's just one more thing to consider when thinking about where radiosensitivity is coming. We can then do the same trick we did in cell culture, use an MDM2 inhibitor. This time we used one that was a new generation developed by Novartis, and we can sustain P53 in irradiated tumors. Now we created a xenograft, we irradiated the cells, and we can sustain P53. And now we can look at the combination of radiation with this MDM2 inhibitor to see the effect on tumor clearance. And this is the results here. We see that in no treatment is the black, we get strong increase in tumor volume. If we just use an MDM2 inhibitor, it's almost as if cells didn't see anything. MDM2 inhibitors seem really promising when they just were discovered, with very low success in the clinic. And I think most of the time because they were used as a single agent. And as you can see, they just don't do much for a lot of reasons that I'm happy to discuss with anyone who is interested. But if we irradiate cells, that we can significantly affect, shrink tumor volume. If we use now radiation plus the MDM2 inhibitor and sustain P53, switch it from oscillatory to sustain, that enhances tumor clearance as well. So what we know so far about in vivo is that the P53 dynamics vary between tissues. The more radioresistant ones show oscillations, the radiosensitive ones show sustained P53. And if we artificially sustain P53 by combining radiation with MDM2 inhibitor, then that can enhance tumor clearance, the clearance of tumors. And again, there are a lot of questions here related to timing and dosing, and a mouse is not a human. How are we translating this to humans? So we're interested in all these questions. I'm just going to at the end share with you a few of our ongoing projects in the lab. We're asking how intrinsic and extrinsic factors shape P53 dynamics. We're looking at the effect of cell cycle phase. Again, it could be differences between cancer cells and normal cells. We're super interested in 3D structures, the spheroids that I mentioned, intestinal organoids, which also can tell us about cell type, looking at differences between stem cells, villi in the crypts, especially in the intestine. How P53 dynamics are translated into downstream processes. As I showed you, we now have new tools to change the frequency, to modulate the frequency of P53 oscillations. We need to remember that when we change the frequency, we also change the amplitude because we have a resonant behavior. And so lower frequency also lower the amplitude. So it might be difficult to say if the effect we see is because of period or because of amplitude, but at least we can start to look downstream and see what exactly cells are reading and how period change affect transcription and also DNA repair. There's some recent studies showing that P53 goes to the site of DNA breaks and physically help with the repair. So that's something we're very interested in. And lastly, in a more kind of translational direction, we're asking, can P53 dynamics be controlled for selective efficient restoration with non-functional P53? If we know that oscillatory P53 is protecting cells and sustained P53 send them to terminal phase, how can we restore P53 in a very accurate and clever way so that we can push cancers with no functional P53 into apoptosis? I just want to mention and thank the people who have done the work. Lots of wonderful collaborators throughout the year. There's some of the people who did previous work I taught. This is the lab that we have right now. I'm not going to lie, Harvard is going through a pretty tough time. The last several months have been, I think, the hardest for the university that I remember in the past 22 years. We've had a very big and vibrant lab that has shrunk significantly. For those who don't know, all our federal grants have been terminated since May, and we're trying to survive. But even given that, this is the lab throughout the years, and the results I've shown you are the combined effort of many, many talented trainees, students, and postdocs. And I keep telling to people that whatever happening, it's really not going to break us. The science is still great. We're having an awesome time. There's things we can't control, things we can, right? And we're focusing on what we can. It's also a note to say that, everyone here, academia is awesome. And as much as I feel like time is difficult and time for research is difficult, this is exactly the time to stay in research, to stay in higher education, to continue science. So just a way of me saying, like, stay in school. Okay. All right. Thank you so much. Happy to take questions.
Okay, thanks for that insightful talk and call to arms at the end. So questions?
Very beautiful talk. Thank you. I have a question. I did not see the expression of gamma H2AX when you actually radiated the cells, and I wonder if there is any correlation between the p53 increase and the expression of gamma H2AX at the site of the breaks, and wonder if that actually has any impact in the cell cycle as well.
Yeah, that's a great question. The question is about gamma H2AX, which is the canonical marker for double-strand breaks. So we look quite a lot at gamma H2AX. The challenge with it is that it's you can only view it with an antibody, and there's no live cell reporter for gamma H2AX because it's the phosphorylation that changes. So we can look at snapshots. What we use is more 53BP1 that localize to the foci, and then we can look at it together with P53. Now, what we see is that when we irradiate cells, there is a distribution of how much double-strand breaks a cell has, a distribution of gamma H2AX, a distribution of 53BP1, right? And there is a very strong relationship between the amount of double-strand breaks and the pulses. So cells that repair faster, in which the number of breaks go below a certain threshold, these are the ones that will stop oscillate. Cells that it take them much longer to repair are the ones that will continue to oscillate. In response to gamma radiation, there's no effect of cell cycle, but in response to other drugs, a bit more messier drug, carboplatin, etoposide, then cell cycle plays a very important role. Yes.
Thank you so much for your talk. So I have a question about the part where you are dividing the P53 oscillations into 3 different phases, zone A and B and C. I am quite surprised that when you perturb the network, right, you see the synchronization of periods so fast. I am, like, at least based on my background in circadian biology, like, usually it takes time before the periods synchronize.
Yes.
Why is there no transition period?
Yes, that's a beautiful question. First of all, we're operating in a very different times than circadian, right? The period, as I said, is between 5.5 hours versus circadian, we're dealing with it, 24 hours. That may explain that. We have in that paper that I mentioned, I'm happy to share copies, all the mathematical explanation for why this is happening and how the cells reset so quickly. But you're absolutely right. And I think what it tells us is that this periodic stimulus that we put is inhibiting MDM2. And even though I showed later that WEAP1 plays a role, then probably other things that play a role, MDM2 really dominate the response. And so we have a knob that is a very strong knob to change that. Now, I would also say it depends on the dose, right? If you add a lower level of the inhibitor, the resetting doesn't happen that quickly and not that beautifully. If you add a lot, then you get a much faster resetting.
Galit, thank you so much for this talk. It was a treat for the morning, and thank you for the message at the end. I completely agree with you. So I'm Marcelo Bonini, I'm from the Moffitt. I have a technical question for you. I'm very interested in how metabolite transients activate gene expression, very similar to what you did with P53. In metabolism, is normally more of this, less of that, and you have your effect. I don't believe in that for a second. I think it's also oscillatory, and the periods will be very important. But we work with ratiometric sensors most of the time. I don't know if you have experience with ratiometric sensors that adds complexity to the mathematics. And if you see any way that this could be used to do very similar calculations as you did with P53, and what are the complexities that we should anticipate when modeling those using ratiometric sensors?
We haven't, so what are you exactly measuring for these ratiometric?
All sorts of things, glucose, reactive oxygen species, acetyl-CoA in organelles. But normally we have a protein that changes fluorescence from one wavelength to another based on concentrations on and off states.
I see.
I think that this will tremendously complicate the modeling, but you know.
We haven't done anything related to that. I think that would be really interesting. And one of the things that comes to mind is something I haven't had a chance to talk about. I think I actually mentioned it in the abstract and then decided to not to include, is that we look here at total P53. But when we look at modified P53, a specific acetylation of P53, it exists in the first pulse, but not in the second pulse, right? So you can look at the ratio between total P53 to acetylated P53, and that changes tremendously between first and second pulse. And we're now looking at downstream what it means. Okay, so if we have tools to do that live, that would be amazing. So, but I should look more into that. Thank you.
Very cool talk. So a couple questions related to the part that I obviously fascinates you and was totally fascinating to me was the 5.5 hour or whatever. Okay, so the fact that that's so consistent is like, one, I have that like existential question is like why, but separate from that, understanding that there are, you didn't speak to it here. Have you looked at all at sex differences in terms of the cell lines and the response? Because there is heterogeneity in like radiosensitivity for sure between the sexes. And there's definitely some role for P53 in P21 toward decisions towards senescence that's been published, but it's out of my grasp.
We have not. We took all the wild type from the NCI 60 cell line. We took all the cell lines that have wild type P53 and measure their behavior in response to radiation. Most of them oscillate, and those that oscillate, it's the same frequency. Some of them don't oscillate, but they have defect in the DNA damage response, ATM, ATR, and so on. None of these studies we pay attention if it's where the cells were taken, right, and the effective gender. So I don't know the answer, but now it makes me want to look into this. Yeah.
Yeah, amazing work. You have elucidated the molecular mechanism, but I'm curious about the physiological logic. Could you at least sort of speculate of what's the physiological reason for the confluency control and for the dynamic threshold for the P53? Why is there kind of higher, the reason that we are sort of evolved the way to behave like this?
Yeah, you need to ask. I don't know, right? These are questions of like, why? I think these are really hard and philosophical questions. Even asking why there is oscillations, we're not entirely sure. We think that that's because, our body have not evolved to cope with 5-10 gray that cancer patient receive in the clinic. We get very small cosmic radiation that leads to a few breaks that usually are resolved within the 5 to 6 hours. So it's almost like the oscillations are a break to not activate apoptosis, not activate senescence too quickly. With regards to why the threshold move, why confluence changed that, I think this is really, really hard. It's hard because sometimes we try to answer why for what we see, but we need to remember it might actually be a side effect or an epiphenomena of something completely different that we're not taking into consideration. So I think at this point, I don't think we can answer it.
Okay, well, I'm going to ask the last question. So one of the things that I'm really interested in is how context potentially impacts how cells behave, who they're talking to, interactions. And one of the beautiful slide that you showed, which with cells that were lighting up with their P53, was it had variability, it had context. So how much do you think the let's say, intrinsic stochasticity that's implicit in the system versus the environmental pressures are more these behaviors?
That's a great question. We haven't looked too much at cell context. I think we're starting to do this now. I think what we will see, but again, this is all to be determined, is that environment is going to have a major impact on the classes of behavior, right? And we're already starting to see that, meaning it will oscillate, it will not oscillate, it will have one pulse, it will be sustained, and so on. And then cell to cell noise or fluctuation will definitely impact things like amplitude, some durations and so on. So we see that there are types of behavior, and then there's a small fluctuation within those classes of behavior. Again, this is all very active research in the lab.
And so would those fluctuations then directly impact whether a cell's going to die or not?
Really good question. What we know, for example, is that when you look at the oscillations of p53, every once in a while there is a cell that probably just because of noise have a series of low amplitude oscillation. And when this happens, those cells may, some of these cells escape cell cycle arrest. Okay, so that's an example in which those stochastic fluctuations can impact what's happening next. But in those cells, p53 dynamics switch to be high. It's almost like a fail-safe mechanism. If you were able to escape the oscillation, now p53 stay high and make sure it doesn't divide. So it's a transient time when the fate is changed and then it stays. With regard to apoptosis, I don't know. And I think, if I understand your question, I feel like that it will be critical to understand kind of the range of how much these fluctuations really matter. Because if they do, then it's a problem, right? Because there's a limit to how much we can control in each individual cell. And then even if we try to control their fate, some crazy cells will do something different. But if they don't matter that much and the environment matter more, then we have tools to gear cells toward particular fates.
Yeah, exactly. So that's what I was thinking. Could you modulate context, and could that actually facilitate pushing them off the cliff, so to speak?
Yes.
Right.
We did to some degree, like we changed things like what we call apoptotic priming, right? So if you change the apoptotic priming, then with small molecules that changes the ratio between pro-apoptotic, anti-apoptotic, they're closer to the apoptotic cliff, and then they fall, even if p53 does the same thing. We look at different cell type in the intestinal organoid, where actually context matter a lot. I don't have an answer yet, but this is exactly what we're looking at. p53 behave differently between these different cell types, and we're trying to link it with their fate.
Okay.
And if any other ideas for how we can look more at context, then I would love to discuss that.
Well, it's crying out for an agent-based model, if you ask me. Anyway, thanks, Galit, for a fantastic presentation.
Thank you.
Right.